I start, I want to acknowledge my grad students who actually collected all the data, Chris and Michael, and my collaborators. What I'm going to talk about today is basically a selection experiment that's been done before. People selected for starvation resistance in Drosophila and melanogaster. You take about 10,000 flies, throw them in a cage, take away their food, and you watch them die. And when most of them are dead, you feed the survivors, you let them breed, and you do this for generation after generation. And we have control populations which are never starved. These are all threefold replicated in this particular experiment. And we're now on generation 85 of selection. And what do you get when you do that? Well, people have shown before you get really fat flies. They have relatively low metabolic rates. If you want to survive starvation, you want to have a lot of energy and use it slowly. They develop slowly. This is because there's a change in their Dyson signaling. They have relatively low fecundity. We've also found recently that they sleep more than control flies. So they sleep about 12 hours a day. And they have bad hearts, and they don't fly very well. And that's what I'm going to talk about today, are these correlated responses to selection. So we've got fat, lazy flies that sleep too much, have bad hearts, and can't do lots of physical types of activities. These are all American flies. They own pickups. They own guns. As a fat human who drives a pickup and owns guns, I really like these flies. Before I go on to talk about their cardiac performance and flight performance, just want to remind you of how insects circulate their hemolymph. They have a heart in the abdomen, which is an open circulatory system. They pump the blood forward, goes into the head, and flows back through hydrostatic pressure. And then they don't use the hemolymph for oxygen transport, for gas exchange. It's primarily the tracheal system. This is an X-ray image. Here are some tracheae. Flies are very active. They also have very large air sacs in their thorax and their heads. That's what's in the purple there. Okay. That's how they do gas exchange. So I'm going to talk about the heart stuff first. And I asked my student Chris for a really good picture of how he does his heart preps. You basically pin a fly on its back with some glue. You cut off the head and half of its thorax. You open up the abdomen, you rip everything out, and just leave the heart behind. Okay? It's kind of like Temple of Doom, except instead of pulling the heart out, you're pulling everything else out. Okay? But the heart's still there, it's still intact. Okay? And so what you can do is then take videos of it. And what we've done here is you take, in this case, the most anterior segment of the heart, anterior region, and you look at a line of pixels down the heart. And as the heart contracts and expands, you can follow those pixels over time. This is a time axis. And you can measure how wide the heart is, or how much it's contracted, how often it beats, all of these things, and extract a variety of physiological parameters, cardiac parameters. Okay. You also did a second technique, which is a little bit less invasive. This is optical coherence tomography. I like to call it echocardiogram for Drosophila, except you use light instead of sound. And but it's basically the same thing. Here you have the flies glued to an agar dish, stuck on an agar dish. You can just line them up and scan them and look at one region of the heart. And over time, you can see the heart contracting, expanding, contracting, expanding. Both techniques give us exactly the same overall results. So what are we going to be looking at? We're going to look at end diastolic diameter when the heart is fully expanded, and systolic diameter when it's contracted, fractional shortening, which you get from those previous two parameters. I'm not going to show you data for heart rate or rhythmicity. Basically, they have the selected lines, the fat lines, and the thin lines have the same heart rates, and at first we thought we found a cardiac arrhythmia in our fat flies, but that disappeared with more data. So I'll just show you data for the first 3 before. All right, cardiac parameters. So, end diastolic diameter is much larger in our starvation-selected flies, and end systolic diameter, when their heart's contracted, is even bigger. The bigger difference between them. The S flies have basically a dilated cardiomyopathy, which is what you see in obese humans. Okay. So you can calculate fractional shortening. How much did the heart contract each heartbeat? In the starvation-selected flies, we only contract about 70%. For controls, normal wild-type flies, it's over 90%. highly significant difference. So their hearts are kind of distended to start with, then they don't contract as much in our fat flies. Okay? So stroke volumes are going to be lower. But you can fix this. You put our flies on a 7-day diet, you take away their food, they become thin, and their hearts recover their function fully. Okay? This also happens in humans. You put the fat person on a diet, as long as it's no other problem that's causing their cardiac malfunction, their hearts will start working better. Okay? These data are from videography. You see the same thing with optical coherence tomography. Okay? So now I want to switch and talk about the flight performance. And this is done by Michael Brewer, a grad student in my lab, who's interested in kinematics of insect flight. And he did a lot of stuff with 3 cameras and even 4 high-speed videos where you can actually trace what the wings are doing with each wing stroke. The data I'm going to show you today, all you have are 2 cameras orthogonal to each other. You're just tracking the fly as it goes through space. Any astronomer can tell you, as long as you have 2 cameras, you can tell where any object is in the universe. Okay? That's what we did with our flies. And what we find, actually these slides are out of order, is that our starvation-selected fat flies fly just as fast as our controls. We're looking at total velocity in 3 dimensions on the upper left. Horizontally, they fly just as fast in the XY plane. However, in the Z-axis, they don't fly as well. They don't climb as well as our fed control flies, our thin flies. They're not good at, they're flying horizontally, but not up. And we think the reason for this has to do with their wing loading. If you look at the mass of our starvation-selected flies, it's 30% more than the controls, and it's all fat. It's not glycogen or other things, it's all fat. These are obese flies. Okay? But if you look at the wing areas aren't different at all. Okay? There's no measurable difference in wing area. So if you look at wing loading, which is the mass divided by the wing area, you find it's a significant difference. These flies are overloaded when they're trying to fly. And if you actually look at the videos, the controls will fly along with sort of a body orientation like this, about a 30-degree angle. If you look at the fat flies, they're like this. So their abdomens are just hanging down, and their butts are dragging, and they can't lift. They can fly sideways, they just can't fly up. So this is very, very cool. And again, you put them on a diet for a week. You take away their food, and they lose their fat. They become the same mass as the flies that are fed controls. And they have the same wing size, because wing size doesn't change when you put a fly on the diet. And they can fly just as well as the control flies. So we found some kind of neat analogies between starvation-selected flies and humans. According to many anthropologists, we've come up with a thirsty genotype hypothesis. A lot of peoples around the world, Pima Indians are a classic example, maybe have gone through very long periods, many generations of low food availability. And the idea is that you put them on a modern European diet, they get really fat because they're not used to being on this diet. Okay? My flies undergo a famine every generation. So, might be analogous, might not be. I'm not saying the mechanisms are the same. You get fat flies, you get fat people, inactivity, both continuous sleep, bad hearts, dilated cardiomyopathy, which is the same thing that you see in obese humans, poor physical performance, and you can cure them by going on the diet. So this is an evolution meeting. So now you're saying, what are the genes? What are the genes? You know, there must be some sort of genes that are causing these flies to be fat and having bad hearts. There are genes that are causing them to be fat. I guarantee you that. Maybe these genes have pleiotropic effects that screw up heart function. Maybe they're linked to genes that affect heart function. Or maybe it's a developmental problem. Maybe it's not genetic. Maybe it's also a developmental problem. Their third instar larval period is a day and a half longer than in their fed controls. They spend an extra 36 hours eating as larvae and becoming fat. They accumulate all of this extra fat. During the larval period, it's caused by a delay in 28-isotope signaling. Maybe something goes wrong with heart development during that extended larval period. Or maybe it's just there's so much fat there that the heart can't beat right. And you put them on a diet, and their hearts are just fine. And we actually have some histology now. We just got it. I haven't actually seen the pictures. But it looks like the fat is actually pressing on the heart. It's actually up under the cuticle, between the heart and the cuticle, and screwing up the aortic muscle function. That's why their hearts don't beat right. So I think it's just, there aren't any heart genes that we can tell. It's just that there's too much fat in the flies. So that's what we think is going on with them. Unfortunately, we were hoping to find the heart genes. Oh well. But you can put them on a diet and they're fine. And finally, I want to acknowledge the people who actually did the work, Chris Hardy and Michael Brewer, and collaborators who helped with all the cardiac types of things. And I'll be happy to take questions.